Hello, we are going to continue with chapter 13 and talk about the stages of meiosis and the various sources of variation which result in very different phenotypes within the. So let's look at the general stages as an overview. So meiosis is preceded by an interphase similar to mitosis. So what happens then is that, remember, during interphase, we have G1, G2, and S phase, not particularly in that order, but S phase is when the chromosomes are duplicated. So remember, the purpose of meiosis is to produce the gametes. So here we have homologous chromosomes. So instead of showing one chromosome, as in mitosis for the general overview, we have two chromosomes, one from the paternal side, or the father, and one from the maternal side, or the mother. The paternal one is blue, and the maternal one is red. Each of them are going to be duplicated during S phase of interphase. And then, of course, during G2, this particular cell here is starting to grow and get ready for the division part of meiosis. Okay, so this is the beginning stages. Now, this is something that's similar between mitosis and meiosis. But after this, it starts getting very different. Now, is one cell division of meiosis that's going to result in still haploid cells with the duplicated chromosomes. Now, this really isn't an accurate representation of what's actually happening here. Each of these cells are going to have both blue and red chromosomes, or should, because there's an event that occurs during meiosis 1 that results in the mixing or crossing over of chromosomes so that they exchange equal segments of DNA. Now, remember the phases of mitosis where the sister chromatids separate, and then, of course, the cells undergo the division. Now, instead of sister chromatids during meiosis 1, it's homologous chromosomes that separate during meiosis 1. Now, meiosis involves two divisions. Remember, the purpose, again, is to produce the gametes. And remember, the gametes have half as much DNA in them, or half as many chromosomes, basically one copy of each chromosome in each of the cells. So, after meiosis 1, after the first division, each of these cells must undergo another division. Is meiosis 2. Now, meiosis 2 is much more similar to mitosis because during meiosis 2, it's sister chromatids that separate during anaphase 2 of meiosis. This results in haploid cells, okay? And basically, these are the gametes right here. Now, again, this really isn't the most accurate representation of the gametes and, of course, the cells that are the products of meiosis 1, because remember, again, I mentioned the mixing up of the chromosomes, and then there's another event that occurs that contributes to this mixing up of chromosomes and increases variation in each of the cells, ultimately, in terms of DNA. So ultimately, each of these four cells are going to be genetically distinct, even though these two cells look like they're genetically similar, and these two cells look like they're genetically similar. They're not, or they should not be. Okay, so all four of these are going to be genetically distinct, and they're all going to be haploid. Now, we're going to go into the stages of meiosis. As I mentioned, meiosis 1 is preceded by an interphase, and the interphase is the same as any other interphase, right? So during G1, the cell starts to grow and starts to prepare for division, right? Or at least duplication of the chromosomes. So once it passes that G1 checkpoint, then the cell can enter S phase, where all the chromosomes will be replicated. And then once it passes S phase, it can enter G2 phase, where the basically the centrosomes are going to be duplicated. And then once it proceeds into cell division, okay, which in this case would be meiosis one, the centrosomes will separate, and then, of course, the uh, spindle apparatus will form, and basically the same thing happens, except there's different things that occur with the chromosomes. Now, division in meiosis one, it occurs in 4 phases. We will not discuss any difference between prophase and metaphase, or like in between, where remember we spoke of prometaphase. So it occurs in 4 phases. So it's going to be prophase, metaphase, anaphase, and telophase. And again, cytokinesis is separate from remember, it's division of the cytoplasm. It's separate from division of the nucleus. So cytokinesis will occur, and the two cells will undergo another division. Okay. Now, during meiosis one, there are a number of things that occur, and most of this stuff occurs during the first phase, prophase one. So synapsis is going to occur. Synapsis is when the homologous chromosomes line up and basically pair up, creating what's called a tetrad. Tetra meaning 4. So tetrad is basically 4 chromatids. Okay, so remember there's going to be 2 chromatids from the paternal chromosome and 2 chromatids from the maternal chromosome. They're going to kind of match up. Okay, so there's protein complexes that allow them to kind of stick together or kind of match up together, synaptimal complex. And they're going to, again, create this tetrad. Okay, so this occurs during prophase one. The next thing that occurs during prophase one is crossing over. So once the homologous chromosomes pair up, okay, they're going to start exchanging non-homologous segments of DNA. So basically, part of the blue chromosomes or chromatids are going to exchange equal segments of between the red chromosome or chromatids. So basically, crossing over is where there are the chromosomes are going to like overlap, okay, and they're going to exchange non-sister chromatids or segments of their non-sister chromatids. They create this X-like structure when they cross over. Okay, so this kind of shows crossing over. Okay, this shows the tetrad, so you can see 2 chromosomes, each with 2 chromatids, so you have 4 chromatids, and then you have crossing over where these non-sister chromatids, so we have a light orange and a dark red one, exchanging in equal segments so that now they're mixed up. Okay, so now this chromosome has part of the, let's say it's the maternal, part of the paternal chromosome, and then this chromosome will have an equal segment from the maternal chromosome. Okay, so when each of the homologous pairs line up, they're going to cross over. And like I said, they form these X-like structures where they're kind of crossing over. These are called chiasmata. Okay, chiasmata. So this is the crossing over and that X shape that you can see when the non-sister chromatids begin to exchange DNA during crossing over. Okay, so again, this occurs during prophase one. So prophase one is where it's at. All this fun stuff is happening during that process. Now let's look at metaphase one. During metaphase one of meiosis, it's the homologous chromosomes that line up at the equator. So remember, we have these homologous chromosomes that are paired up, okay, and they create this tetrad. At this point, they have already crossed over. So during metaphase one, as you can see, we have the homologous chromosomes lining up, and we can see that these 2 pairs, the red is going to be above the equator, and the blues are going to be below the equator. And then, of course, this one shows the blue above the equator and the red below the equator. So there are 2 ways in which they can line up at the equator. And remember, there are multiple homologous chromosomes that are going to be lined up at the equator. So this cell only shows 3, but human cells are going to show 23 pairs of chromosomes that are going to line up at the equator. So that is a lot of more potential variation here. So you can have different combinations. And why it contributes to variation is because they're each going to be separated into their own cells, creating genetically different cells, even more so than just the crossing over. Okay, this is called independent assortment. Okay, and this occurs during prophase 1 when the homologous chromosomes line up at the equator. And remember, all of this process, I'm just talking about the new things, but remember, you should know that, remember, the centrosomes have separated, the spindle apparatus is formed, the microtubules had polymerized, there are non-kinetochore microtubules, and there are kinetochore microtubules that are going to bind at the centromere of each of the chromosomes, or homologous chromosomes. So all the other same events which allow cell division to occur are going to occur similar to that in mitosis. Okay, so let's talk about anaphase. Okay, so anaphase 1, it's the homologous chromosomes that are going to be separated during meiosis. So you can see that the red ones are going to end up in this cell, and the blue ones for these two are going to end up in this cell, and then the blue one for this one is going to end up in the cell with the two mostly red ones, and then the red one from this pair is going to end up in the cell with mostly blue ones. So you can see there are multiple different new combinations that can occur the more sets of homologous chromosomes that exist in a cell. Phase, it's the same process, right? So now we have two cells. These are animal cells, so a cleavage furrow is formed from constriction of the cytoskeleton. And then, of course, the nuclei are going to reform. Then what's going to happen is each of these two cells from meiosis 1 are going to undergo another cell division. However, this next cell division is not preceded by an interphase. Okay, so division in meiosis 2 occurs in similar 4 phases, except they are they have the Roman numerals 2 associated with them. So instead of prophase 1, we have prophase 2. Okay, and then of course metaphase 2, anaphase 2, and telophase 2, until we end up with the 4 gametes that we're going to have after both divisions have occurred. Now, like I said before, meiosis, mitosis. So let's go through this process. So here we have during prophase 2, of course, the centrosomes are separating, the spindle apparatus is forming. We have non-kinetochore microtubules versus kinetochore microtubules, right? So again, these are all chromosomes here with individual chromatids. And then the chromosomes are going to line up at the equator. Remember, it's not homologous chromosomes during this stage. It's chromosomes similar to mitosis, except these chromosomes are genetically different. Okay, so they're just basically mixed up. Okay, so by the time metaphase 2 occurs, the centrosomes are on opposite poles of the cell. You can see the non-kinetochore microtubules versus the kinetochore microtubules. And then what's going to happen? Anaphase 2, and this is where sister chromatids separate. Okay, so similar to mitosis, here it's sister chromatids separate, and different from meiosis 1, it's sister chromatids separate as opposed to homologous chromosomes that separate during meiosis 1. You're going to end up with 4 cells. Each of these cells are going to be haploid. They really kind of started out haploid at the point where meiosis 2 begins, or they're considered that way. And they each have 1 copy of each of the chromosomes, and then they're going to be individual cells. And these are the gametes. So let's compare mitosis to meiosis. Now, I've been doing this as I went through the stages of meiosis, but it's kind of better to put it all together. Mitosis is basically just cloning, right? So it's very similar to what would be considered asexual reproduction, and it is an asexual process. It's just basically allows the growth of an organism, okay, or just basically an increase in cell number with the same number of chromosomes. So it conserves the chromosomes. And this is as opposed to the meiosis. Meiosis reduces the number of chromosome sets. In humans, we start out as diploid. Okay, so remember our somatic cells are diploid, and then the somatic reproductive cells are going to undergo meiosis to produce the gametes, which are haploid. Now, there are 3 events that are unique to meiosis, okay, and all of them occur in meiosis 1. So remember synapsis, when the homologous chromosomes line up and match up, and then crossing over between the non-sister chromatids within the homologous chromosomes. This occurs in prophase 1. And then the homologous pairs of chromosomes are going to line up at the equator of the cell, creating the metaphase plate. Well, there's different ways in which they can line up. Remember, there's 2 ways in which they can line up. Okay, so that increases some variation. And then separation of the homologs. Okay, so it's homologous chromosomes that are separated, not sister chromatids during meiosis 1. Okay, so comparing meiosis to mitosis, right? So we have 1 division. We start out with 1 cell. You end up with 2 cells that are genetically identical to each other and to the parent ultimately. Okay, and then, this is the parent. And then we have a parent cell here. Now, during meiosis, after duplication, we have homologous chromosomes that are going to line up, and then they're going to kind of mix up, right? So during crossing over, which occurs in meiosis 1, and then they're going to line up at the equator, and there's 2 ways that they can line up. Okay, so in terms of colors, the blue and red could either end up on the left or the right, which would eventually be in different cells after the anaphase occurs. And then we end up with 2 cells that are considered haploid at this point at the end of meiosis 1, and then each of them undergo another cell division, which is meiosis 2, which is most similar to mitosis, creating haploid cells. This is a good table showing the summary in terms of each of these processes. I'm not going to read all of this. This could be something that you might want to rewrite, maybe in your own words to help you study. But the properties that are considered comparing the mitosis and meiosis is DNA replication when it occurs, the number of divisions, whether synapsis of homologous chromosomes occur or not, and then a number of daughter cells, which are the result after the division, and their genetic composition, meaning are they identical or are they genetically distinct from one another? And then, of course, the role or the function of each of these processes in animal cells. Okay, so on our final note, we're going to talk about sources of variation. Have you ever thought to yourself when you saw perhaps families that some of the children don't look like the parents, or one or more of the children looks more like the father or looks more like the mother, um, and they are, the children, right? So how does this happen? How do we get so unique? Well, it's because of 3 sources of variation. And I've touched on a couple of them as I went through the stages of meiosis. So one source of variation comes from independent assortment. Remember when I touched on, when we talked about the metaphase and metaphase 1 of meiosis and metaphase 2, there are different combinations of ways that these chromosomes can line up at the equator. Okay, so when the homologous chromosomes or homologs separates during anaphase 1, there can be different combinations that result in those particular cells. And remember, they've already crossed over, and I haven't talked about crossing over yet. However, that is another form of source of variation. Okay, so this is independent assortment. Basically, the homologs are going to assort differently when they line up at the equator. Now, in terms of independent assortment, let's talk about numbers, right? So how does this relate to differences among individuals, even within the same families? So it's really the number of combinations that are possible when these chromosomes independently and randomly assort at the equator. So if you think about it, there's two ways that the pairs of chromosomes can line up at the equator. Okay. So here we have the number two. And really there's 2 to the n different combinations per individual. Okay, so n is the haploid number. So remember, there's going to be 23 pairs of chromosomes lining up at the equator. Okay, now 2 to the n represents the number of variations for one individual. Okay, and then another individual can contribute to the n possible combinations of variation. And then combining the two, that's basically 2 to the n plus n. So that's 2 to the 46 different combinations upon a mating that can result in variation, contribute to the variation. This is part of how we become so different, how we have so many differences. Okay, so when you kind of put the numbers to it, we have 2 to the 23 times 2 to the 23, which would be 2 to the 46 different possible combinations of chromosomes as they line up at the equator. Now that's one source of variation. The other source we've talked about, crossing over. This one's probably the most obvious source of variation, right? So remember, crossing over produces what's called a recombinant. So recombining, the chromosomes have recombined. So they basically mixed up. So they exchange equal segments after they form these chiasmata, or here's a single chiasma. Okay, that's like the X that forms when the non-sister chromatids overlap, and then they exchange segments. Okay, so then you end up with mixed chromosomes during meiosis 1. Okay, so then the sister chromatids of those mixed chromosomes are going to separate during meiosis 2. Okay, and then they'll end up in, at the end of meiosis 2, they'll end up in individual cells, and you can see all the gametes are genetically different. So this is just one small fraction of the differences between the gametes that can contribute to the next generation. So remember, we have gametes, we have the eggs and sperm. So remember, all of the billions of eggs are going to be genetically different, different combinations of all these chromosomes, which underwent all these changes. Okay, and then of course, same thing with the sperm. Okay, so there's different combinations of all these different changes and mixing up of genes and alleles so that we have these different gametes. Now, you have billions of choices of sperm and billions choices of eggs. Which ones come together increases even more variation. Together, this is called random fertilization. So it's somewhat random as to which sperm ends up meeting the egg, and which egg it is, and then once they fuse, now it's a combination of mom and dad. So mom and dad had their own gametes, which had different combinations of their own DNA, and of course their ancestors. And then you combine mom and dad to create a whole new organism, which is a combination of 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 genes from 2 different families, right? So now we can have all kinds of differences, and this kind of represents this is Waldo, where's Waldo kind of idea here, but this kind of represents the number of differences between each individual, and how unique we could be just because of the variation that occurs during meiosis, and then of course random fertilization. I discussed the stages of meiosis 1 and meiosis 2. You should all remember what is involved during interphase when we discussed that in the chapter on mitosis. Remember, interphase only occurs once during meiosis, before meiosis 1, and then there are 2 divisions resulting in 4 haploid cells. Now, during this process in meiosis 1, there are two sources of variation coming from meiosis 1, and then random fertilization is totally different. Okay, so remember, there's sources of variation in meiosis 1. They include independent random assortment. Remember, assorting the chromosomes at the equator during metaphase 1. And then crossing over occurs during prophase 1. And then, of course, random fertilization is going to occur after the gametes have been produced, and it's random as to which gamete is going to meet up with the other gametes, right? So we have egg and sperm, and they fuse. All right, thank you for listening.